those who aren't yet aware, I am Dr. Aaron Wyman. I am Associate Faculty of Biology here at Spring Arbor, and I have been looking forward to this day for 5 months. They have not, but I have been. Because today, we had 8 students presenting work they did over a summer. Let me put this into context for you. Many of the programs they applied to had somewhere between 500 to 800 applicants. 10 were selected. We are awesome. When I say we, I mean you. You are awesome. Don't let anybody tell you different. I had more fun this summer at the meeting that Maddie and Mary are going to talk to you about, because I was talking to faculty, like, you had how many students selected for some research? I said, like 8. How many bio majors do you have? Total? Maybe 75. And they turn ash white because they don't know how we're doing it. And when I told them it's God, they're not sure what to do with that. But unfortunately for them, they're dealing with me, so I told them how that works, because I'm subtle, like a sledgehammer. All of you can do this. Every one of you can do this. You'll keep your hair on like me, but you can do this. I am so proud of these ladies. They know that. All of us are proud of these ladies. And they are going to give you a short bit of what they did this summer. We will post longer versions of their individual talks up to what's called the internet in the next bit in terms of time. Afterward, they're available for Q&A. Please ask them questions. I know I'm a frightening man. I'm 5'7", bald, 43 with acne. I am exceptionally frightening. I get that. Talk to them. They are none of those things. It's okay. All right, I'm going to lead off with Rachel. Rachel's going to tell you work she did this summer at Michigan State.
So like Dr. Wyman said, my name is Rachel, and I worked at Michigan State in the Takahashi lab this summer. And I did work using Arabidopsis thaliana, which is a plant that biologists use often. And my project was titled The Impact of Cli-Clavato-1 Signaling on Root Gravitropism. So first, it's important to understand that there are different types of nitrogen within the soil. So there's nitrate, which is found in the lower strata of the soil, and there's ammonium, which is found in the upper strata of the soil. Plants prefer nitrate, so they seek to elongate their root system architecture, so they lengthen their root to reach that nitrate at lower strata of the soil. So to do that, plants have to have signaling pathways that allow them to gauge the amount of nitrogen within the soil and how much farther down they need to go to achieve more nitrogen, and so on and so forth. So to do that, they use different pathways, one of which is the Cli-Clavato-1 pathway, where low nitrogen induces the expression of Cli3, which is a small signaling peptide, which interacts with Clavato1, a kinase, which phosphorylates downstream components to then inhibit lateral root primordia, which would produce these side roots. And it also has an impact on gravitropic response. So gravitropic response is this idea where a plant can respond to gravity by different signaling mechanisms that I'm not going to explain. But we found that this Cli-Clavato-1 pathway also played a role in gravitropic response. So that's what I studied this summer. And this here, so my lab hypothesized that PLBP1, phospholipid binding protein 1, induced that gravitropic response. So I did a lot of studies over 10 weeks using root gravitropic assays that I describe in my video that's going to be posted. But basically, I measured the rate at which roots would turn in response to gravity over a 9-hour period of time. And what I found was that wild type, called Col0, in potassium chloride was our negative, where there was no nitrogen, it induced gravitropic response. So it turned stronger, more strongly. And then under ammonium and nitrate application, there was similar gravitropic response. But in PLBP1, there was an overall lower gravitropic response. So this is a knockout mutant. So there is no PLBP1 in the plant. And so it can't use that in the pathway here, it can't use that to induce gravitropic response, so it's lower among all conditions. So in conclusion, I did that with other genes as well and other work, and that allowed us to develop this model where long distance nitrogen inhibits PLDP1 expression and low nitrogen induces this pathway that is ammonia dependent as well. But we found that long distance nitrogen signals weaken root gravitropic response overall because that nitrogen does not require the plant to grow anymore into the soil, so it stops that growth. And that the loss of PLDP1 causes agravitopic roots. So I would like to acknowledge my lab that I worked with, seen here, minus one member who was out of the country. Specifically Katerina Lay, who was right here. She was my mentor and helped me with a lot of this. I was in the Takahashi lab. And Dr. Cornelius Berry was our program director and my funding. Thanks.
Hi, I'm Ashley. This summer I worked at Mississippi State University doing transition metal complexes for olefin aziridination and cyclopropionation. So I know those seem like really big words. So what I did is I was developing aziridines and cyclopropanes. So an aziridine is a 3-membered cyclic ring with 2 carbons and a nitrogen, and a cyclopropane is a 3-membered cyclic ring with 3 carbons. So I wanted to talk to you guys a little bit about what the REU is. So the biggest thing about the REU is that it prepares students for grad school. So if you plan to go to grad school, doing an REU is really helpful because it just gives you an idea of what it kind of is like to be a grad student. It also gives you hands-on lab experience with equipment that you might not be able to use here, because I know that a lot of the things I use we don't have here. And it helps you network with different professors at other universities, and it also teaches students how to write manuscripts, which is also really important if you want to go to grad school. So I got a lot of training while I was there on some stuff that we don't necessarily do here. We do rotary evaporation here, but I never knew how to do that, so that was really cool to learn how to do. We also do prep to seed, which if you've taken organic or you're inorganic, you know what that is, but this is like what you do on steroids. It takes anywhere from an hour to 3 hours to develop instead of the 10 to 15 minutes, and it's not really the most fun. It just takes forever, and then you have to sit there and watch it and make sure it doesn't develop too far. It's not very fun. I also learned how to do X-ray crystallography, which, as you saw on my first slide, I have these crystal structures up here of the catalysts that I was using. So that's how we were able to determine what the catalyst looked like, was with X-ray crystallography. And then I also learned how to use NMR, which is something else that you would learn how to interpret in organic. So I worked with synthesizing aziridines a lot more than I did with the cyclopropanes. So this is kind of what our reaction looked like. We did a lot of different olefins, so different alkene derivatives and styrene derivatives to make our 3-membered cyclic ring with nitrogen. So these are the different catalysts that I used throughout the summer. The first is copper fen. So it's a copper with 2 phenanthroline derivatives on it. The second is copper dioxofen, so a copper with 2 phenanthrolines that have ketones. And then copper DHOP, so copper with 2 phenanthrolines with epoxides, and silver fen, so a silver with 2 phenanthrolines. So there's also something else I did that wasn't really intentional. I kind of accidentally discovered a new molecule while I was there. I don't really know how it happened. It was a side product of one of my reactions. It's called trisc-fen copper 1 triflate. So a lot of copper complexes, they form octahedral when they have copper 2, but this was a copper 1 complex, so normally they don't have 6 things bonded to them. So in the world before, there were 4 known octahedral copper 1 complexes, and I accidentally discovered a 5th. So I don't really know how. So how we did it is I was running a cyclopropionation reaction, and then we wanted to do some gas chromatography on it, and we dissolved our product in diethyl ether, and we went to take the GC, and I guess you can't really see it, but there's like little black dots in the bottom of that vial, and those are crystals. So yeah, we really don't know how it happened as of yet, but we've been trying to reproduce it, and before I left, they hadn't yet been able to do it other than as a side product. So, yeah, that's what I did this summer.
Good morning. My name is Tori Buckles, and this summer I worked at the AuSable Institute of Environmental Studies. My project, reforestation of vacant oil pads in northern Lower Michigan, aimed to restore the fragmented forest left behind by oil drilling activity. In the past, when oil and gas companies vacated oil sites, they were only required to move their extraction equipment. Reforestation efforts were not required as the sites were expected to naturally reforest, and this was not the case. Although grasses and other herbaceous vegetation did reestablish on the sites, most remained barren of woody vegetation even 30 years after extraction had ceased. Today, energy companies are required to follow a vegetative restoration plan set in place by the Department of Natural Resources, but still struggle to reforest these areas. Of the 60,000 oil and gas-related wells in Michigan, I studied 9 in Antrim, Kalkaska, and Otsego counties. The AuSable Institute, indicated by the blue star in the figure on the right, was in the heart of the oil drilling activity. This gave us a unique opportunity to study something that directly impacted the local community. I was the 4th year in a 5-year ongoing study. In 2015, 3,600 trees were planted, 900 of red pine, jack pine, red oak, and white oak. Then 3 active surface treatments were then applied: disking, fertilizer and disking, and a control treatment. To determine the best methods for reforesting vacated oil pads, we then found the growth and survivorship of each tree species. We also collected soil samples and vegetation surveys using a Daubenmeier frame. We found that disking produced the highest survivorship and highest growth in every species and every year. Disking enhanced survivorship of trees by reducing competition, by stimulating root growth in seedlings, by aerating and breaking up the soil. In contrast, fertilizer actually decreased growth by increasing competition with herbaceous vegetation surrounding the trees. We found that pines experienced positive growth in all treatments, while oaks experienced decreased growth due to herbivory by white-tailed deer. There were no evidence of soil toxicity on the pads or limitation of macro and micronutrients, although they did differ in soil characteristics. Non-native species were present on all sites. They actually accounted for 50% of the total plant cover. We found that the control treatment was actually the one that actually was affected least by the non-native species, leading us to determine that treatments actually increase non-native species rather than decrease them, which answered one of the fundamental questions of our study. Further recommendations include adding wood chips along with the disc treatment. This was an accidental discovery. On one of our sites, there was an influx of volunteer jack pine and a dramatic increase in height. This was because there was actually an old logging deck present on the site where felled trees were stacked, and when we applied the treatments, they were then disked into the soil, and this caused a dramatic increase in jack pine. Forest managers who want to mitigate site-specific deforestation on oil pads should plant red pine on disc soil, and if they are going to plant oaks, then they should include mesh barriers to protect the trees. This project was a real blessing to me. I gained experience in the field, collecting data, analyzing it, and presenting it. I was nervous at first to live in the middle of the woods because that's where AuSable is located, but I made some great friends out of my coworkers. I saw wildlife up close, sometimes too close, but that's okay. AuSable taught me how to care for God's creation by healing something that had been broken for so many years. I would greatly encourage anybody who has the opportunity to pursue research to do that outside or outside campus, and to experience what it means to do hands-on research. Thank you.
Hi, I'm Jessica Walker, and this summer I was at Worcester Polytechnic Institute in Worcester, Massachusetts. And the project that I was working on all summer was the thermal stability of phosphoinositide domains. So this is the phosphoinositide family, and the one that I specifically worked with this summer was PI45P2, otherwise known as PIP2. And the reason why we looked at this was because it has a negatively charged head group, and you can interact with positively charged amino acids along with multivalent cations like magnesium and calcium. And this is the data that wound up being the best I collected the entire summer, with different percentages of POPC, cholesterol, PIP2, and then my donor fluorophore and acceptor fluorophore, which was top fluor PC and Litipi TMR PIP2. So this was taken at an expectation of 470 nanometers, and this right here is the donor peak, and then this right here is the acceptor peak, and we increased the temperature by 10 degrees, starting at 20, ending at 60, and this worked out the best. We did try starting at 20 and increasing by 5, but there wasn't much of a difference between the peaks for the acceptor fluorophore. And then what I wanted to talk to you guys about for REUs was the things that you do outside of the lab for REUs. So it's a lot of fun. You make great friends with like all of them. I actually still talk to quite a few of the people that I met at my REU. And the first thing that we did was go to Purgatory Chasm, which was near the college, and it's just this chasm that you can climb through and walk through, and it was pretty fun. I didn't like it that much because I don't like climbing that much. And then we also did the Freedom Trail in Boston, Massachusetts, which was a lot of fun. We didn't walk the entire thing, but we did see a majority of it, and we went past the cemetery that had a lot of like founding fathers, like all that, the people who first came here, and we didn't actually go into it, but because it was so full. But it was an interesting thing to just see. And then the last thing that we, as a group, kind of chose to do, away from like the professor's scheduled activities, was we did a lot of escape rooms, and that's what this picture right here is. It was a lot of fun to do that, and it was just a great experience overall to have.
Hi, I'm Natalie. I'm a senior here at Spring Arbor and a biology major, and this past summer I spent 10 weeks in Charlottesville, Virginia at the University of Virginia School of Medicine doing research on human papillomavirus and how it can cause cancer. And I'm going to talk a little bit more about my program in general, because if you're more interested in the research, we did film videos which are very research heavy, and you can check that out. But what was really interesting and unique about this program is it was really geared towards people who wanted to do biomedical research. So about half of the students in my program wanted to go on and become doctors, they wanted MDs, and half of us were interested in PhDs. And there were a few people interested in MD PhDs, which is a little crazy to me. Yeah, so it was very much geared towards biomedical research. So we got to talk with a lot of doctors, a lot of residents. We had weekly seminars about up and coming biomedical research, and then we spent pretty much all of our time, 40 hours a week, working in our labs, working with whatever our specific question was. And my question had to do with the human papillomavirus and the specific protein that it creates called HPV16E6. This is a high-risk type of human papillomavirus, and it can cause cancer, which is why we were interested in it. And we wanted to know if through associating with this pathway, if it stops the action of a tumor suppressor or something that helps your cells not become cancerous. So to answer that, I got to do a lot of different tests this summer, which I've never done before. So now that I'm in genetics, I'm doing a little bit more of these, but I never really worked with protein assays before, or DNA assays, and this is called a Western blot. It's a type of test where you can look at proteins and where they are. And, I got the results from this from an immunoprecipitation experiment that I did, which is where you take antibodies and they kind of fish out certain proteins of interest that you have, and you can see what's attached to that protein. But I got to learn a lot of techniques this summer that I'd never done. We did a lot with cloning, which is essentially creating your own DNA sequences, which I didn't even know you could do. And then I also got to learn tissue culture, which is a really specialized field of research where you grow cells in a plate and you can add things that you want to them. You can put the DNA that you cloned inside of them and see what happens, see what proteins they make. You can put antibiotics on them and see how it affects it. So that was really cool, I guess, to use those skills that I've never learned before. The first day when I stepped in the lab, I didn't know how to do anything besides, the basic lab work. But by the end of the program, at the end of the 10 weeks, I was designing my own experiments and, showing my PI the results, and it was really cool to see it come full circle, everything I learned throughout the summer. But one of my favorite parts about the summer, as interesting as the lab work was my peers and getting to hang out with the other people in the program. I lived in an apartment with 3 other girls, and everybody on the floor of our apartment was in the program. The program actually paid for travel to and from Charlottesville. They paid for the housing over the summer, and we all got a pretty generous stipend. So the only thing you really had to account for yourself was food, but the stipend definitely covered that. Virginia is a beautiful state. I'd never been there before, so I got to do a lot of hiking in the Shenandoah National Park. I went to DC. That picture on the bottom left is from the Smithsonian. Downtown Charlottesville is super fun, so I had a lot of good times with my other people in the program. It was more than just research the whole summer. It honestly felt like a vacation when we weren't in the lab, so that was really cool. And I just want to acknowledge Dr. Vanderpool, the head of my lab, and then Nicole Breimer, who's the lab tech. And she showed me everything that I worked on this summer. And they took a lot of time out of their own schedules to be really patient with me and teach me everything that they had learned. And the organizers of the program for sure, and then also just my professors at Spring Arbor. I applied to 12 different programs, so there were a lot of recommendations that I needed, and all of them were more than ready to write those for me whenever I came to them with a new place that I was interested in. And one important thing I learned over the summer was, even though schools like University of Virginia, they're really big, they have a lot of money, what my professors here taught me as far as critical thinking and then just learning how to move around a lab, that's what was most important for me, and you can go anywhere if you have that. So I place a lot more value on Spring Arbor than I did before, even though I already did before, so it's been a really cool experience to look back on, but just be really thankful for what we have here. So.
Hi, my name is April Kriebel, and I spent this summer at Auburn University, which is in Alabama, and I was in the lab of Dr. Rita Graves. And what we studied was transcription factor binding site diversity in Drosophila. Now, for those of you who don't know, the common name of the Drosophila is just a little itty-bitty fruit fly, and they make a great model organism. And this is because you can maintain large populations with relative ease, which means you have high power for all of your statistical testing. Then you also have a high number of offspring and a short development span, which means that genetically you can cycle through a lot of generations very fast. They also, even though they only have 4 chromosomes, they're conserved to the human genome, which means we can take what we know and learn about fruit flies and use that in studies of humans. So what are transcription factor binding sites? So when you have a strand of DNA, there is what's called transcription factors, and the binding of those transcription factors at a transcription factor binding site is going to regulate the expressivity level of your gene. Now, the reason those are hard to study is because it's not like a turn-off light switch. It's not completely on or off. It's like on a spectrum, and so that makes studying them really hard. And so we wanted to look specifically at 2 different signaling pathways controlled by transcription factor binding sites. And that's the FOXO and the acetosome signaling receptors pathways. And those have both been linked to secondary sex characteristics in flies. And secondary sex characteristics are just characteristics that develop as a result of your sex. So that would be like physiological, like mating behaviors, etc. So the concentration of my REU specifically was to use Temple, which is a computer software, to assess differences in transcription factor binding sites between various populations of fruit flies. And the 3 populations that we specifically chose to focus on, one was from Africa, one was from Ithaca, New York, and then one was from California. And the reason we chose those 3 population sites was because the fruit fly is believed to have originated in Africa, and we wanted to look at the change in the genetics as the Drosophila spread out, because it's supposed to have followed human expansion as well, so we wanted to see if the genetic changes would follow that correlation. But how it fit into the larger research of the lab was that we wanted to take the differences that we found this summer, and then the lab will continue our work by doing population genomic analysis to identify the signatures of adaptive evolution. And then they'll take those differences and they'll find differences in phenotypes, and then they'll evaluate those phenotypes to see whether those express sexual differentiation. So our results were mostly in FST, and FST is a factor that we use to measure how different the two genomes were. And so our results were, as hypothesized, Ithaca and California, the two populations from the United States, had the closest conservation to each other in terms of the transcription factor binding sites. The Rwanda and Ithaca and the Rwanda and California showed slightly higher diversity, which we would expect. So our hypothesis matched our conclusion, but for our questions of further interest, we had decided to identify genes that express differentiation strongly between the populations. And so these are just examples of genes that were highly variant in the two US populations, and these are variations that were high between the US and Africa populations. And what we can do with those genes is, now that we've identified them, we can explore them, look at their human homologs and those effects, and then we can explore their connection to the insulin signaling pathway, which has a lot of applications for human research as well. So that was what we concentrated on research-wise. But like the ladies before me have said, we just had a lot of fun. Like, I would recommend doing an REU, like, not only for the lab experience, but also for like the friendships that you form. Like, I was in a dorm with Auburn had a bunch of different REUs, so they had like physics students and stuff like that, so you get to hang out with people who aren't in your particular subsection. But also, like, the people in your subsection are amazing, and we did a lot of cool trips. Like, Auburn isn't that far from Atlanta, so we went to Atlanta. We saw like the bodies exhibit down there. I don't know if you guys have heard of that. We had a weekend in Panama City Beach. That was really fun. Got some beach time. Me and my roommate went like blueberry picking, bonded over our mutual love of avocados. Like, it was a great summer. Also, I hadn't had a whole lot of lab experience going into this summer. And so just my mentor, Dr. Graves, was incredible. But also the undergraduates and graduate students who she already had working in her lab, they gave me like my first like in-depth experience with lab culture, and they could not have been like more welcoming, more understanding of the fact that I didn't have like a very deep bio background. They answered all my questions, and like I left that summer like feeling a lot more confident in my ability to interact with the sciences, and just like present a more solid and well-rounded application to graduate school. So it's a really good summer. Thank you.
My name is Maddie. I'm Meredith. And this summer, well actually the past year, we've been doing independent research here at Spring Arbor. And then this summer we were able to take that research and present it at the 2018 American Society of Plant Biologists in Montreal, Canada. And we looked at the influence of ginkgo leaf extracts on fungal and bacteria growth. And so what we actually did was we took this paper by Wong, and we said, this looks like an interesting topic. They take ginkgotides and they see the effects that it has on fungus. And we thought, one, this looks really interesting. Two, we want to see if we can use this protocol in microbiology to make a new experiment, to come up with a new lab, because we're always looking for creative ways to use things like this. So I worked with the fungus. This is Fusarium oxysporum, and it is a common tomato pathogen. And we worked with several different assays. This was the one that worked best. This was the one that they actually used in the paper. And what we did was you take a plate of potato dextrose agar, you punch a hole in it, and then you put the fungus in that hole and you let it grow. And on each of these pieces of filter paper, you have a different level of extract. So you have 100%, so on and so forth, different dilutions. And then we have our controls of the sterile water and the glacial acetic acid, because the ginkgotide extract is kept in the glacial acetic acid, which has a pH of 4, so we wanted to make sure the pH wasn't killing anything. And we observed this crescent shape we're reading about here, and that just showed that the ginkgotide extract was actually having an effect on the fungal growth, and we showed exactly what the paper showed. So that was promising to know that we were doing what Wong was doing in his paper. We also looked at spore viability, which was a little bit more complicated, but we said, I wonder, we know it has an effect on the growth of the fungus. Now, does it kill any spores? So we took spores, we put them in solution, we added different levels of ginkgotide solution, and then we dyed them, and we looked to see how much dye was left after it was rinsed. And that the more dye, I believe it was the more dye, the more spores. And here you can see that the higher the ginkgotide extract, the fewer spores were left. They were not viable, which means they were not able to procreate. And we used this using a plate reader, and we looked at the absorbance value, and that's just how we read how many spores were left. And my research led into Meredith's research. She worked with bacteria.
So as Mary said, there was published research on how ginkgo tides affect fungal growth, but there was no published research on how ginkgo tides affect bacterial growth, or even if they do. So that's what we decided to look at for mine. So what we did is we cultured bacteria in wells. The wells had varying concentrations of the ginkgo tide solution, and then we also used a known control of sodium salicylate, which we had done previous research with, and then water. And so as you can see, some of the wells are darker. That indicates a higher bacterial growth because we stained them, all of the wells, and so then the stain would then dye any bacteria that was left. So with the higher concentrations of ginkgo tide solution, there's very little to no dye or coloration, and that's because there was little to no bacteria to be stained. But then in that 0% ginkgo tide extract, the color was much darker. This is just quantitative data of the same information. Again, you can see that at the 0% solution, there is much higher value. That's because, again, there was bacteria present that was stained, so when we read the absorbance values, more light was absorbed by those different concentrations. So while we were in Montreal, we didn't just go to, we did go to the conference. At the conference,
He made us go to the conference.
We did go to the conference. We got to share our research with different people, a lot of professors at other universities across the country, postdocs, graduate students, and it was really cool to interact with them, and they were really eager to learn about what we were doing, our research, and everything like that. Then aside from going to the conference, we got to explore Montreal and the city and some of the history. And eat food and just see different sites, the city, and a lot of the history of the influence of the French.
And that was my last trip anywhere. All right. So, do only pre-med students do these? Do only ecologists? Well, the UV plant people. Is everybody available? So anyone can do this any number of times. I'm going to let you all start asking them questions, and understand that if you don't ask questions, Dr. Baldwin and I will have to ask questions, and they would prefer that you ask them. Amen.
Accidental discoveries. And I'm wondering if you have any comments about the role of accidental discoveries in the development of science.
Basically, all of science is an accidental discovery.
Well, Rachel disagrees.
All of chemistry is an accidental discovery.
Yeah.
I didn't say that all science and all scientific things are discovered by accident. I think that we take systematic approaches with understanding of the basic elements of our research, and sometimes we don't find what we thought we were going to find, and in that way there's an accidental discovery of something that you didn't anticipate, and you flush that out further through your research if that's interesting to you. So for my project, they found PLBP1 from an old basically survey of expression levels in different types of plants, and they found that this was different consistently among different mutants, and so they wanted to look into that. And so that's how they developed a significant portion of my project, as well as publications that came from my lab. But it stemmed from something that was intentionally chosen. But I mean, sometimes wood chips end up being tilted in soil.
Yeah, that was really interesting. I actually went to a meeting that a lot of the MDNR, there's a whole oil pad, basically association that comes from Ensemble, Huron Pines, another nonprofit. They came together to discuss this problem because it is a problem. There are 60,000 oil and gas sites, and they're basically just huge fields filled with nothing. But the wood chips were an accident, but they did come up in this meeting, so this accidental discovery, basically at the end of this 5-year project, is going to make a big impact. They're looking for ways to reforest, and that accidental discovery can make a huge impact on reforestation in Michigan.
I know some of you guys kind of touched on how much experience you've had previously, but can you guys kind of talk a little bit more about individually how much experience you guys had beforehand? Because I know a lot of people are worried about that when applying.
So I'll just start. I had a fair amount of experience. I had more than some in my program, but less than others. But I have been doing independent research here for like 2.5 years, and I did this RAU. So I would be more on the upper end of having experience. But part of that is being in a smaller institution that it's limiting in a sense. I can still do a lot of research here, but there's an upper limit, and that's where going to an RAU helps, because they have money and grants for equipment that cost, several $100,000. Like, we don't have that grant for that. So, but I would say that I had a fair amount of research experience.
I mean, going into applying, I didn't have much. I had like maybe half a semester of research experience, like independent research experience, like into applying. I mean, into going, I had about a semester and a half, but still, it wasn't very much experience that I had going into it, other than working in lab. So.
I thought that I didn't have much experience going into it because I thought, the research that we did in classes was maybe different than the type of research that they did at bigger schools, but really what I had done in my even just, basic biology and chemistry classes were super valuable for what I did this summer. And one thing I was really worried about was, are the other students going to have tons of experience? And I'm not. But there were even students in my program from really big schools. One girl was from Michigan State. And I didn't feel like my research level or my experience level was any different than what they had. If anything, I think, I kind of said it in my presentation too, but I feel like us science students at Spring Arbor, we really know how to critically think differently than some other schools do. So I think that was really helpful. Yeah, not really. Not a ton, at least outside of school. The one thing that I think appealed in my interview was that I did research in class in genetics for the Arabidopsis thaliana, and that got me a position basically at Ensemble studying trees. So there are transferable skills, but I guess I impressed with what I had.
And I had no independent research. I just had the labs for the classes. So you do send your transcript in, and they do look at that pretty thoroughly. But what was kind of interesting for me was I was in a biochemistry lab, and I'm taking biochemistry right now. So they do teach you a lot of what you need to know for that specific project.
For Mandy and I, it was a little different because we were doing independent research and not in RAU, but I think I speak for both of us when I say that our experience was limited to doing labs for different classes here in high school, which is not much.
I joined the biology party super late because I was a mathematics major, and I still am. I just wanted to take a more bio approach to it. And so last summer I spent at the University of Minnesota, and I had no experience doing any research. And so I think that if you're in that position and you're applying for REUs, a helpful thing is apply to as many as you can. I think I applied to 18 last summer, 18 programs, and then just make sure that you have you're asking faculty you have a solid relationship with to write your recommendation letters, because coming from a small school and having a personal relationship with them can really make you stand out. And so then coming into this summer, I think that helped a lot, having Minnesota. But I didn't have a whole lot of research experience coming in.
Yeah, Meredith kind of covered me. This was my introduction to research. It was here, let's teach you everything you need to know right now. So I learned a lot from this experience, and basic things like streaking a plate to running a plate reader. You learn a lot while you're doing it. So you get thrown into the work and see what you can do.
Just like to hear all of your, a rough estimate of your acceptance rate, how many schools you applied to and how many you got into. I think that's helpful. I think that'd be helpful for folks to know.
Well, I applied to, so part of how REUs work is they have different application dates. So a long time ago, they all had the same application date, and they all would tell you at the same time, so you could get into 3 and you pick one of your 3. But now you you applied to this one February 5th, this one February 12th, and this one March 1st. So by the time you've applied to March 1st, your February 5th gets back to you, but March 1st hasn't read your application yet. So I got accepted to Michigan State University, and I had a unique situation where I needed to be within a certain geography. And so I got married this summer, and so I needed to be able to like not have to drive 4.5 hours to do something. So I needed to be like within 4 hours-ish. So Michigan State kind of worked perfectly for that, and so it was Michigan State or University of Michigan was my top preference, and MSU got back to me first, and one in the hand is worth two in the bush, so I took the one I got.
So I applied to 22 RUs. I got rejected to 16 before I even got an acceptance, so that was kind of really depressing. But then I got waitlisted on one, I got into 2, and then 2 of them I never heard back from. But I mean, so it does seem kind of depressing at first, because like I didn't even get accepted until like a little more than a month before I left to go to my RU. But don't be discouraged if you get rejected to 16.
I applied to 12 programs. I got into 4, got rejected from maybe like 2 or 3, and then the other ones I didn't hear back from because I'd already accepted somewhere else. So I applied to 1. Okay, to be fair, I applied to one position. I wanted to do macroinvertebrates, but they were like, you can't do that because we've accepted another person. He was really cool, anyway. But we have another position for you if you're willing. And he was like, do you know what an oil pad is? And I said, no. He said, do you want the job? And I said, okay. So my experience.
I applied to 15, got waitlisted for 2 of them, and the one that I actually was accepted to, that application wasn't due until March 1st, and then maybe a week later, they emailed me and said that I was accepted. And so I had to start emailing people, like Rachel did, being like, so how's the application process going? And then just wound up going to that one and didn't hear from a lot of them.
I applied to 0, got into 0, so I mean you could say that's 100%.
My first year, I think I applied to like I said, it was a lot. It was like 18 and I got into 1. And then this year, I applied to, I think, similar situation to Rachel's a little bit. I started applications for like 12. Auburn got back to me first, like I accepted going there. And then I ended up getting into another one, but I'd already accepted at Auburn, and then some of them I didn't finish. So I'd say I probably like finished applying to like 10 or 11 and got into 2.
I didn't apply to an REU. Dr. Wyman said, do you want to go to a meeting in Canada? And I said, Canada sounds like fun. And so I did research and went to Canada.
Will you guys apply? Do you know like what you're going to be researching, or is it more blind?
Well, at least in my case, like for most of the ones that I applied to, like there is a list of like different projects, and you could pick like 3 to put on your application. So like I got one of the projects that I put on my application for Mississippi State, but for the other one that I got into, they didn't tell me what project I was going to be working on. They're like, you'll find out when you get here, and I was like, no. But so I mean like sometimes you know what you're gonna get, but then other times you don't. So I mean, some of the applications didn't even have like a list of potential projects.
I would say most had a list. They would have a list of faculty that have traditionally been involved in REU programs, and you can read what they study and what you could anticipate working with. When I received the acceptance for Michigan State, they didn't tell me what lab I was in, and I didn't find out for a while. And I didn't get one of the 3 that I picked, but I ended up in a biochemistry lab, and I talked to the director because he was with us every week, so we were talking about how people got placed into what lab, and he said basically like, I have a focus in molecular biology. He's like, I can put you in any lab. There are people who, when they do coding and when they do computational biology, they can't go into just any lab, when that's their expertise. And so I ended up in the lab I did because of my general skill in every area that they can put me in.
I have a question on your research, actually, Meredith. I was wondering, where are you going to go with that now? So did you prove that sodium salicylate inhibits less than the ginkgo extract?
That is what it appears. However, we're still like working on that, and we're gonna do more tests with like different bacterial species just to see. Okay, maybe it only works for like these 9 or whatever that we tested, but maybe it works for like a wider array. So just testing different species.
And I noticed, like on your results, there were certain species, like E. coli, for example, that didn't have any results.
Yeah, it just knocked it out pretty much completely.
They completely. Okay. Thank you.
I want to ask you what your grades were while you were doing research. Did they care? Number 2. Rachel and I had a discussion many times. Rachel, what was more important? What classes you took or who you knew?
Who you knew, my goodness. I didn't have an appreciation. So it always, it's who you know, it's somewhat what you know, and it's more who you know. And I didn't really have an appreciation for that until I went to Michigan State, and I realized that people in my program got there because this wasn't always the case, but some people in my program got there because people they knew at their home institution knew people at Michigan State. And those were different situations because the lab that they were working at Michigan State funded them specifically, but they were still able to get there because of that personal connection. So they were able to make thousands of dollars and get a 10-week research experience to put on their resume because they had personal connections and they had done well in the labs, and they had, exercised critical thinking and shown, hey, I'm a person that you really want to send to your collaborator, and who you knew.
So you're saying that it doesn't hurt to make good relationships with your current faculty? That would be useful. You all have heard this now. You should talk to your faculty. Like eyes, not at the feet.
Hey, Dr. Wine, I got some questions.
Yeah, I'm up here. Other thing we learned, and I was fascinated with, they want sophomores. They want sophomores. They're okay with you being sophomores. You can take it however you want. But the reality is they want sophomores. So if you are a sophomore, you've had the audacity to take Organic 1, and you're going to take Organic 2, that's a good choice, by the way, and you've taken genetics, roll the dice. The worst they're going to say is no. Same true of dating, it turns out. But nonetheless, the worst they're going to say is no. Any other questions? Give them the hand. They are awesome.